Okay, so we are happy to have with us today Dr. Luciana Regitano. Luciana Regitano, she graduated in veterinary science at the Federal University of Paraná, Brazil, obtaining her master and PhD in genetics and breeding at the Superior School of Agriculture Luiz de Queiroz of the University of São Paulo, and she joined Embrapa as a research in December 1994, where she's the leader of the Animal Biotechnology Group at Embrapa Southeast Cattle Research Center, and she collaborates with the Genetic and Evolution Department of the Federal University of São Carlos as an invited professor since 1997, where she teaches molecular genetics applied to animal breeding. With experience in genomics throughout her career, she has made important contributions to the development and application of molecular markers in animal genetic improvement, with special emphasis on selection for production in tropical environments. She's currently dedicated to understanding the functional relationship between the bovine genome and the genomes of microorganisms, exploring the concept of hologenome for the development of technologies aimed to more sustainable livestock. Thank you for being with us, and the floor is yours.
Thank you so much, Samala. Thank you for this invitation and for the opportunity to talk about the work we are conducting in Brazil. It's very nice to be in touch with you and be able to interact. So I will talk today about this, let's say, this challenge. Could the Bos indicus hologenome be considered for breeding purposes? So as it was said, I work for Embrapa, which is the agriculture research service in Brazil. Just for you who don't know much about Embrapa, we have a number of research centers. I'm not sure if you can see my pointer here.
Yes, we can.
Okay, thank you. So we have 43 research centers and 7 central units which deals with administration. My unit is this one here in Southeast Brazil. It's the Embrapa Southeast Livestock. And most of our units are dedicated either to products, to agricultural products, or to environments. So at my unit of Embrapa, we have an experimental farm of 2,500 hectares. And so it's a huge farm right on the border of the city. And we have today 132 employees, from whom 40 are researchers. And our research is mainly on livestock production systems, plant breeding for grazing systems, for feeding animals. And the Animal Biotechnology Lab, it started on 1997 when I came from my PhD to return to my position. And we've been dealing with genomics of beef cattle, tropical adaptation, beef quality, and sustainability traits. We joined the post-graduation program on evolutionary genetics and molecular biology at the Federal University of São Carlos since 1997. And it's been a wonderful collaboration since then. I get many students, many undergrad and grad students from the university, and this has been very fruitful. Why we are focusing on beef cattle? Because Brazil has a very important role in the world beef market. These are data from 2020, but it didn't change much from there. And we usually have the largest herd and commercial herd. In 2020, we were 217 million herd. We always are among the 3 major world producers of beef. And we are first in world beef export. We were in 2020, but we are usually among the 3 first exporters in the world. Brazilian beef is mainly based on Bos indicus breeds, especially on the Nelore breed. And this represents around 90% of the beef cattle herd in Brazil, either as a pure breed or crossbred. 80% of this production is based on grazing systems. And we have around 168 million hectares of grassland. So it represents around 45% of the Brazilian territory. I'm including here in this map not only the cultivated grasslands, but also the natural grasslands in Brazil. The feedlot practice is not very usual. We have around 21% of our cattle being finished in feedlots, but it's growing at a rate at 6% per year. One of the big problems nowadays with cattle production, with livestock in general, is the production of greenhouse gas. As bovine are ruminant animals, they have a number of microorganisms in the rumen. They have a stomach divided into compartments. And in this rumen, in this compartment called rumen, we have a number of microorganisms that produce most of the nutrients that will be absorbed by the animal, by the gut, but also the fermentation of the feed would release CH4, methane, which is considered the second global greenhouse gas, representing 15 to 20% of the global greenhouse gases. And 7 to 18% of this methane is produced by livestock. Well, we know also that when digesting the plant material by bacterial fermentation, we produce methane, which means that this carbon will not be used by the animal. So there is a dietary loss, energy loss of 2 to 12% from the production of methane. It's releasing carbon to the atmosphere instead of absorbing and getting this carbon into the production system. Methane has a medium to high correlation to dry matter intake, whereas the correlation to feed efficiency is very hard to derive. There are papers saying that there is a correlation. Some works are showing a favorable correlation. Others are showing unfavorable correlations. But in many cases, the correlations, the magnitude of this correlation is not very high. Well, feed efficiency is also a very important trait for cattle production. We have feed representing up to 40% of feedlot costs. And when we think about having an animal who's more efficient on using the energy of the feed, we can consider that by having these more efficient animals, we will prevent the expansion of land occupation, because we don't have either to open more grazing areas or to use more land to produce the feed for animals that are kept in feedlots. The other point is that, as I told, we have a correlation with methane emission that could be selected as an indirect response to selection. So we consider that both methane emission and feed efficiency are sustainability traits. Why these sustainability traits are not selected or improved in routine in Brazil? Well, mainly because to analyze, to score these phenotypes, we have to use automated feeding systems, or in the case of methane, we could both use a yoke, a handmade yoke, that you have to have the animal holding this handmade yoke in his neck. And you can imagine that not all animals are happy with that. And so it is very hard to have this animal in the pasture or hanging around with this yoke in his neck. And the other possibility is to use automated gas traps. But in both cases, there are problems of investment. And if we consider a cattle herd of millions of animals, how can we implement this type of evaluation in a routine? So our group started a long time ago a multi-omics approach to develop biomarkers and to understand the relationship of genome, the functional genome, and these traits. Especially at that time, we were working with feed efficiency. And so we had a huge experiment with 796 steers, from which we collected 45 phenotypes. And we had the genotypes in high density SNPs. We had also 20 sires with their whole genome sequenced in Illumina short reads. We also collected muscle and liver, 200 samples of muscle, and we did mRNA-seq and miRNA-seq. And we also did proteomics from 65 of these muscle samples. And by integrating this information, exploring this information, we were able to find the regions of the genome associated to feed efficiency. And we described a reasonable irritability and genetic variation for feed efficiency, which was not known for Nelore. So we pointed out that it would be feasible to select Nelore for feed efficiency. But when we did this GWAS for feed efficiency, what was very clear was that in Nelore, residual feed intake was truly polygenic. There was no evidence at all for major genes. When analyzing the functional genome, the RNA-seq data from muscle and liver, we also found genes that were differentially expressed between high and low feed efficiency animals, both in liver and muscle. And we found a number of these genes as differentially expressed in liver and muscle. And what was interesting is that most of these genes, when you do an enrichment analysis, you see that genes for lipid oxidation and ketogenesis, as well as genes for oxidative stress, were enriched in the animals that were less efficient. Well, another interesting result from the integration of SNPs with expression data was the EQTL analysis. We found, this was the work by Aline Cesar, and we found
Okay, so like, okay, so can anyone type on the chat if it's only me that lost Luciana or is everyone?
I can't hear either, Samla.
Oh, thank you. Well, let's wait for a little bit. She will probably be back.
I'm sorry, we had
No,
power.
it's okay.
Yeah, we had a power shutdown. Let me just see here. Okay, let's go back then for the EQTL results, right? So we had these interesting results showing that we have many SNPs in the genome that regulate the expression of mRNA. I don't know how much familiar you are with this kind of analysis, but in this circle plot, what you find here, each of these bars is one of the Nelore chromosomes, chromosome 1, 2, and so on. And each of these points represent one SNP that is connected to other SNPs, other transcripts that are in other positions of the genome. So we are looking at transregulatory SNPs that, if you look at these 3 points here that are highlighted, which are EQTL hotspots, you will see that they are transcription factors. So very nice, and we also saw that when you make a superposition of these eQTLs with bovine QTLs, if you apply a test for random overlap, you will see that it's not random. So in fact, the QTLs, the bovine QTLs, are enriched for eQTLs. So we also did a similar study integrating SNPs and allele-specific expression. Sometimes we have one of the two alleles of one SNP not being expressed or being expressed in a lesser amount. And this indicates that close by we should have a ASE QTL, which is another SNP that influences the rate of transcription of one allele. In this example here, we have one transcription factor that binds in this position and transcribes this segment that contains the SNP. And once you have this sequence here, this C in this position, you don't have the binding of the transcription factor, then this allele is not transcribed. Are you hearing me well?
Yes.
Okay, thank you. The other study we also did with this data was to find specific SNPs in specific regulatory elements, like this work by Tainan Cardoso, a postdoc in our group, who analyzed the presence of SNPs in genes that code for transcription factor and transcription cofactors. And she found one SNP in this transcription cofactor who is associated with muscle gene expression and also with residual feed intake in Nelore cattle. As you can see in this figure here, the EF1A1 is a central element for the expression of many genes, and it is correlated to residual feed intake. Well, but remember I told you that the less efficient animals had genes that were related, had more expression of genes related to oxidative stress. And then once I was in this ASAS meeting, and I found this very interesting talk about from Lelu Guan from the University of Alberta, saying that she found less efficient animals enriched for free radicals producing microbiota in the rumen. So this was quite a coincidence. And I asked myself, could gene expression be related to the oxidative stress that's produced in the rumen? So could we see this difference in gene expression in Nelore because they are facing more free radicals produced by the microbiome as described by Guan? And while this idea was pursuing me and I started reading this literature about microbiome, and then I was much more amazed to see that there are many works saying that host genetics and the rumen microbiome jointly associate with methane emission, or that host genetics influence the rumen microbiota, and there is an irritable rumen microbial feature, and this could be associated to feed efficiency. And well, and then you start seeing that there is epigenetic regulation of the host genome by the microbiome, and that most probably the non-coding RNAs are the elements that regulate this crosstalk between host and microbiome. And so you step by new concepts like the microbiability, which is defined as the proportion of the phenotypic variation explained by the microbiome. So could we think about deriving breeding values or microbiome values for the animals? And also, when we think about this crosstalk between the genome and the cattle genome and the symbiotic bacteria and microorganisms, and how one regulates the function of the genome from each other, could we think about the hologenome as one unit of selection? So we started thinking what could be the contribution of a multi-omics approach to this matter. And we thought that by using a multi-omics approach, again, we could think about models for genomic selection once we have the additive effects of this microbial composition. We could find causative mutations or SNPs that are related to differences in microbiome composition. We could find targets for gene editing or RNA silencing, biomarkers, and even pharmaceutical candidates. So when at this time we had in my research unity, there was this project, the Water Footprint Project, that was coordinated by a colleague, Julio Palhares, and they were investigating whether using byproducts diet would reduce the water footprint of cattle production. And they had this experiment with 26 animals in a regular diet and 26 animals in a byproducts diet. And they were collecting phenotypes that were really important for our project, like feed efficiency and methane emission, as well as water consumption, individual water consumption. So why not taking a hire on this project as a pilot to start understanding this relationship between cattle and microbiome? So we collected from these animals, we collected ruminal content and stool, ruminal wall, liver, and muscle. And this is how science goes in Brazil. First, we were able to approve a small money from the National Council of Science in Brazil, which was only enough for doing the 1618S sequencing and metabolite profile. And then we approved a much bigger budget from the State of São Paulo Foundation, FAPESP, and we started the Nelore hologenome, the real one, where we could add metabolites, mineral content, metagenomics, shotgun metagenomics, metatranscriptomics for the rumen and stool content. And we also did RNA-seq, miRNA-seq from the host tissues as well as proteomics. We are dealing at this moment on collecting the proteomics. All the other data is collected already. So when we started analyzing the metabarcoding, the 16S sequencing of these animals, we found that one of the points we would really like to prove is that instead of using rumen, we could use stool samples because it's much easier to collect. So in a large scale, collecting rumen samples is impractical. And what was really interesting at this first moment was to find that rumen and stool would share 96 bacteria and 47 archaea. And the most important finding of this study was that the methanogenic archaea were shared between rumen and stool samples. and mainly because it's very probable that reasonable to think that the fecal methanogen population depends on the rumen. It's not actually colonizing the latest portions of the intestine, but it's being carried with the matter from rumen. And in this second work, we showed that these amplicon sequence variants could be related to methane emission and feed deficiency, both in rumen and stool. So here we see for feed deficiency, in these graphs, we have the feed deficiency in the Y and the abundance of ASVs. And we can see that we have ASVs in the rumen that increase the RFI, and also one ASV that is related to decreased RFI. We're not actually thinking about cause or effect, but they are associated to increased RFI or associated to reduced RFI. And when we come to methane, we also have in rumen one ASV and in stool another ASV associated to methane. And what's interesting is that these two bacteria found associated to residual methane emission and residual feed efficiency, feed intake, were bacteria that are supposed to have anti-inflammatory actions, effects over the gut system. And it's been shown in many studies that inflammation can be a cause of reduced efficiency. Well, when dealing with the shotgun sequencing, this is the work, I'm sorry, the previous was the work by Bruno Andrade, who's now a professor at the University of Munster in Cork. And this is the work by Liliane Conteville, who's dealing with the shotgun sequencing of feces and rumen. So we can see here from her results that there are differences when considering diets, both in feces and rumen. And because she's dealing with whole shotgun sequencing, she's now able to mount the genomes of bacteria and archaeas that are present in the rumen. Of course, these are metagenome assembled genomes, which means they are not exact genomes. Most of them might be chimeras, but it's still a very good source of information. And she was able to mount 887 genomes belonging to bacterial sequences and 29 genomes belonging to archaeal sequences. And she found that the individual diversity of the animals was negative correlated both with residual feed intake and residual methane emission. So the most efficient and less emitter animals will have a higher diversity, both in rumen and stool. This other work by Tainá Cardoso is, she ran an algorithm that relates, that searches for epistatic interactions between SNPs and phenotypes in general. So in here, what you can see, she did a model trait correlation plot. So in the A part of the figure, you have the modules of SNPs that are correlated to each other. So each color here is a group of SNPs that is correlated to each other and is correlated to RFI. And in this B part of the figure, you see the modules of SNPs that are correlated to each other and are correlated to residual methane emission. And she plotted both RFI and RME in the two figures, so we can see that the modules that are correlated to RFI are not correlated to residual methane emission. And the same here. So we are seeing that these two traits regarding this specific analysis, they behave as independent. The SNPs that are associated to one trait are not associated to the other. And in the C part of the figure here, what we see is from each of these modules that are associated to RFI, how many SNPs are associated to phenotypes, to metabolites, to archaea, to bacterial ASV in feces, to archaea and bacterial ASV in rumen, and to mRNAs in rumen. In the case of this experiment, we just have functional data, RNA-seq and miRNA-seq from rumen samples. Unfortunately, when we started collecting the samples from these animals, the first sample we collected were stool samples, and we didn't realize we could do it in a way to preserve RNA. So sad. But anyhow, we're dealing with this information in a different population right now. So we already have mRNA and miRNA from stool samples from another project. And so here what we see is that all these elements are interconnected. And we see SNPs correlated to residual methane emission trait when you take the genes that are around these SNPs. We see that they are related to ion transport, glutamate receptor signaling pathway, RAC1 GTPase cycle, and ERBB2 signaling. Whereas the SNPs correlated to residual feed intake are related to different pathways, different metabolic pathways like calcium-dependent events, regulation of cation transmembrane transport, which was also seen in our, these metabolic pathways were also seen in our previous studies with host functional genome. So this project is based on a very small sample size. So we decided we should go further and get information from a larger population to start trying to prove that this information is valid in a large scale, in a population scale. So we started this second project, which is the Nelore's microbiability for feed efficiency, reactivity, and beef quality. We started collecting stool samples from the animals that are submitted to the National Young Sire Evaluation Test from the Brazilian Association of Zebu Breeders. So this association makes a sire evaluation test every year with around 120 Nelores and animals from other Zebu breeds too, but at this moment we are only focusing on Nelore to have to focus, because it's such a huge data and it's such intricate information. And this project is financially supported also by the Brazilian Association of Zebu Breeders and by the National Council of Science. And this is very preliminary work result. Before we had the budget to analyze all these stool samples in 16S sequencing, we sequenced only the extremes of the RFI distribution in the first year of evaluation testing, the animals that were evaluated in 2019. And we compared high and low residual feed intake animals. And again, we see that these more efficient animals here have higher alpha diversity, which means higher individual diversity associated to efficiency. The great question is how to model this microbiome in selection. Can we derive models to include the information of microbiome on to select animals for different traits? I didn't mention, but there is a large literature about the relationship of microbiome and brain development, as well as psychological traits. So in humans and in mice, they are showing a high relationship between microbiome and psychological traits. And that's why we're dealing here in this project also with the reactivity of the animals. So how do we deal with this problem? How do we introduce this new layer of information on breeding? There is this work by Alejandro Saborio Monteiro in livestock science, where they used simulation data to check whether introducing similarity matrices between microorganism communities in the hosts would be a way to deal with this information. And they compared 4 models: the classical genetic model, GBLUP, a model where only microbiome was considered, the model with genetic and microbiome, genetic microbiome plus genetic microbiome interaction. And what they saw is that this 4th model here, the complete model, including not only the information of microbiome, but also the interaction of genetic and microbiome, would be a best fit, would increase the accuracy of prediction. And while this is quite obvious when we think about everything I told you till now, what we see on that epistatic relationship and so on. But could we think about other ways of using this information? Like, could we use microbiome parameters as indirect phenotypes? For instance, could we use diversity index, individual or bulk taxa associated to phenotype? And such a thing like a microbiome-assisted selection. Could we consider SNPs associated to microbiome features in genomic selection? I hope to have these answers for you in few years from now. And so just to finish this presentation, I would like to say that there are evidences of microbiome influence on sustainability traits in Nelore. And we see that stool samples are useful for microbiome prospection, so this is a good news. And we need to think about methods to incorporate the microbiome information in selection. I really hope to thank all this wonderful team who's been working in these 3 projects for a long time together. And it's such a large and nice team, many wonderful people working together, and many institutions involved, not to forget to thank the financial support from FAPESP, CNPq, CAPES, who's responsible for a number of scholarships in Brazil, and the Brazilian Zebu Breeders Association. Thank you so much for this opportunity again, and I'm very happy to take questions.
Well, thank you for your presentation. It was amazing, how much content. And if anyone has questions, you can raise your hands, you can turn on your microphones and camera and ask your question. So I do have a question. I was thinking about if eventually we end up using this for selection, I was thinking like, I believe that if we change the diets of these animals, this microbiome and the fact that will change. And then I was thinking how we could like pass by these and understand like how much I predict for a group of animals that the microbiome could help in in a few traits. So if I change the diet, how this would affect my final result?
Yeah, this is a very important question, and I'm not sure if I have an answer for that. I think that we will think about something like a permanent environment, which is the basis of the microbiome of the animal, but sure, the diet has to be taken into account. So you will be always dealing with an environment by microbiome interaction, and you will also be looking at perhaps a triple interaction: genome, environment, and microbiome. So this has to be considered for sure when dealing with selecting animals that are more efficient. Like we are also looking for the microbiome of animals under grazing systems, and to check whether the microbiome could be a good predictor for feedlot. But it's a very complex question.
Yeah, I can imagine. Okay, so anyone has questions? Do you believe we will be able to use microbiome for genetic prediction in the future?
Well, I think we will, but perhaps more than genetic prediction, the microbiome will be important for building modulating strategies. For sure, we can select animals that have a better microbiome, or at least select against the ones that have dysbiosis. Mm-hmm. But I think that the more useful information will be for developing biotechniques and pharmaceuticals or nutraceuticals that could help us modulating this microbiome.
I see. Perfect. Okay, so Flavio, you can go.
Hi, Luciana.
Hi, Flavio.
How are you? Good to see you.
Good to see you too.
Yeah, thanks a lot for the presentation. Very interesting. I have a kind of maybe a comment and a question for you. Okay. I remember I read some research done that what they did, they get some cows and they removed completely the rumen, okay, and then they allowed naturally to recover the flora and fauna of the rumen. And what they note that the composition before and after was very similar, right? So it means that the host is kind of determined that composition. So in this sense, I always think about this. Would not be really better for sake of selection to select the host only and bring then the microbiome that's associated with the genomics of that host, right?
Mm-hmm.
Because as Samuel mentioned, then if we include in the evaluation the microbiome, then you have this huge interaction with the environment. That
Mm-hmm.
I don't know, I didn't see any proof that this is going to lead to higher genetic improvement for the traits that we are looking at. Can you comment this? What is your view on this?
Yeah, I totally agree. This is a very interesting work, and they've done this with mice also, the defaunation of the intestine, and that's where they started to pursue the information on heritability of these traits, because clearly the host genome is modulating the composition of the microbiome. So this would be a point to in favor of selecting animals with a better microbiome. The great point is selecting a better microbiome for which situation, like Samela was asking. So this has to be considered for sure. Are we selecting animals for high grain? Are we selected animals for grazing systems? Or how this microbiome will behave from grazing to high grain? And sure, there is, and it's been like in the work I showed you from Tainan, there are SNPs associated with the trait, with this composition. So we could think about using these SNPs on selecting a better microbiome, and even using the microbiome as a phenotype to be selected.
Mm-hmm. Yeah, I just wonder if you select the host that you are not going for that particular situation, for that, let's say, diet, or you are not bringing also the best microbiome.
Mm-hmm.
Right? So
Mm-hmm.
that's what I like to see more results, maybe to be convinced
Yeah.
that talk about selection, right? Not feeding strategies and other things like pharmaceutical applications.
Mm-hmm. Yeah. Yeah. There are some work in swine, in pigs, showing that there is response to selection to microbiome, and the inclusion of microbiome would increase accuracy of breeding estimates. But pigs are quite different, right? They have a much more stable environment, controlled environment than cattle. And they are not ruminants, so it's quite different from cattle. But I think that some of the information we can, and even when you use the microbiome relationship matrices, there are points that you have to consider, because if you build a matrix, that I was just discussing this with Daniela Lourenco, and this is when you have a matrix of similarity, when the difference are so small, because the basis of the microbiome is very similar across animals. So we would have to think about another way of considering this relationship, perhaps only dissimilarity matrix instead of similarity.
Yeah. And there is another aspect that complicates, I think, is that you need to discuss and think about which is the proper weight that you're going to give to the host and to microbiome to achieve the highest progress, but medium long term, not short term, right?
Yes.
I think I didn't see any study on this. So that is new.
Yeah, it's a
Lots of
very
things to
open
do, right?
field.
Very
Yeah,
open field. It's very interesting. So,
very, very.
Luciana, thank you so much for participating today. It was really
You're
very
very
nice.
welcome, Flavio,
Thanks.
always.
Yeah. Bye now.
Bye.
Okay, so we just have two questions on the chat from Bruno. So he asked if in the case of breeding for more efficient animals, would we select animals able to host efficient microorganisms?
I would say so. I would say so, because there is a good association of microbiome and efficiency. So I would say that the selection for feed efficiency should select for better microorganisms as a correlated trait.
Okay, and he also asked if could we supplement these animals with better microorganisms?
Yes, yes. The point is, as we were discussing just a little before, the host will always try to put the microbiome to its original configuration. So whenever you are using a probiotic strategy, you have to use this probiotic strategy always as a continuous treatment. There are many research in human and mice showing that better than probiotics is the use of prebiotics. So if you feed the animals with feed that is good for the microbiome, favors the good microbiome, then you have better and more lasting results than when you do probiotics.
Okay, very well. Well, if anyone has one last question.
Thank you, Bruno.
Okay, so, well, would like to thank you again for your talk. It was very nice to have you here, and hope to have you again in the future, maybe with more update results. And I would also like to thank everyone that watched us today, and next Friday we'll be here again waiting for everyone.
Thank you, Samala. It was my pleasure.
Okay, bye.
Bye.